Well, good evening. It is 7 o'clock now, so let's start because we have a lot to cover today. I'm delighted to have with me today Professor Brigitte König at this AONM, that's Academy of Nutritional Medicine webinar. She runs a laboratory in Germany and does excellent mitochondrial testing as well as other kinds of tests. And she's been involved with the mitochondria for more than 20 years. She'll maybe tell us a little bit more about herself as we proceed in the webinar, but I won't go into her bio in detail because it was in the description that you received of this webinar. So we're going to talk today about the mitochondria and chronic health conditions. We do have a chat button at the bottom if you'd like to put any questions or comments in there, or in the Q&A, and we'll go through as many of those as we can at the end. We'll probably talk for 40 or 45 minutes. We have a huge number of charts, so some of them we won't talk about in detail, but you will receive all of them as a chart pack afterwards. And this is only part 1, because there's so much to say. So there'll be a part 2 in mid-January, and doubtless some further webinars in the course of next year as well. So I'll just pull up the slides now. So we'll start by just mentioning that there are webinars about the mitochondria on our webpage, aonm.org, and then mitochondria, and then mitochondrial webinars. So if you'd like to know more about the mitochondria themselves, that's all described there. They are the powerhouses of our cells. And you can see from this wonderful film here made by Harvard professors that actually there are far more than 2 or 3 mitochondria in each cell. In some cells, there are hundreds, in some thousands, and in some, quite literally, millions. So they do a lot more than just give us our energy. They have 2 sort of energy production engines in them. One is called the Krebs cycle. One is called the electron transport chain. And we will primarily be talking about the electron transport chain inside the mitochondria when we discuss one test today, but we'll always identify which part we're talking about. It has 5 complexes, and we'll be explaining a bit more about that in a moment. 36 ATP are generated along the electron transport chain from glucose, and far more from fats. 146 in the case of oleic acid, approximately. Of course, it varies hugely. That's if you can let fats into your mitochondria. And we'll be looking at one of the tests that Professor König does in her laboratory that shows what fuels your mitochondria are able to metabolize. And you can see from her tests that fats aren't always able to get in. So it's an incredibly complex process, and lots more about this to be found in, for example, this wonderful article that is referenced at the bottom of the page.
Gillian, can I just mention you're not sharing your slides?
Oh, I'm sorry. How stupid. It's very important that I do that. Apologies. So let me just go back to the beginning. And am I sharing them now?
Yes.
Okay. So I'll go back to the beginning, but I won't talk about them in detail. I'll just, or should I, Claire? Should I start again?
You can go briefly, whatever's best for you, Gillian.
Okay. Well, just to say that we'll be talking today about the mitochondria and chronic health conditions, and our mitochondria power our cells and a lot more, as you will know from other presentations that you've seen of ours. The electron transport chain inside the mitochondria has 5 complexes, and that's the key engine for producing our currency that gives us energy, called adenosine triphosphate. And you can make 36 of these energy currencies from glucose, approximately, if the mitochondria are able to work from glucose. But if you can let fats into your mitochondria, then you can produce far more ATP. 146 is what this slide is telling us, but each fat has its own ability to produce this adenosine triphosphate. And now we should be able to produce our body weight in adenosine triphosphate every day. And so that's an incredible amount. Imagine if you weigh 70 kilos, that's 70 kilos of ATP. And Professor Nick Lane, who spoke last week at London's mitochondrial conference organized by the Nutritional Medicine Institute, explained that gram per gram, you're converting about 10,000 times more energy than the sun every second in your mitochondria. So that's really incredible. That's again, if they're working well. So what do we lose if our mitochondria aren't functioning optimally? Bioenergetics, which is what we'll primarily be talking about today, is the high energy or high intensity energy metabolism and nutrient sensing. That will not work ideally. Biosynthesis, they also do, and that means we won't be making heme optimally or metabolizing urea or cholesterol and other nutrients as well. And then signaling a lot of different functions. What we felt is that perhaps we'll do a separate set of webinars, if you would like, if there is appetite for it, on these individual aspects next year, because there's so much to say. Imagine that the mitochondria are responsible for releasing cytochrome C, which you need to be able to do to get rid of cells that are not functioning well, like in cancer. And that apoptosis process breaks down if your mitochondria aren't working optimally. So these are the conditions and many more linked to mitochondrial dysfunction, with references after each. A big thank you to Dr. Joseph Pizzorno, who last weekend spoke about this in detail. So do look up those slides if you're a practitioner and would like to access them from the NMI, because they are all available too. And so now I'll turn over to Professor König to talk about the pathways that we'll be covering today that may have become compromised in mitochondrial dysfunction.
Yeah, hello. Yes, there are different possibilities. Some of the energy genetic pathways that may become compromised. We talk today about the macronutrients and the micronutrients, the cofactors. And first is the question, are there enough macronutrients to fuel, to give the cell sufficiently materials? And are they the correct ones, efficient for generating mitochondrial energy? That is the macronutrients. And sure, we have also to look at the cofactors and the micronutrients. Are there the correct substrates in the Krebs cycle? Are the mitochondria able to metabolize the substrate? And if not, what is the problem? And especially if there are some problems, we look at the complexes of the electron transport chain and whether they have the right substrates or whether they have the right composition or the right function. I think that is where we focus today. Gilian, ich höre dich nicht.
Sorry, I'm not functioning well today, am I? Need to look at my mitochondria. Apologies. We are talking next time about the other aspects, like how many mitochondria there are in the cell and the oxidative stress and the obstacles. That we will mention some obstacles right now. So one thing to look at before you even start, there we are. So this is the ATP profile. Important, Brigitte, and not everybody knows, is that this is the mitochondria at rest, isn't it?
Yes, it is.
Could you tell us about this? Thank you.
It is the ATP production, or the ATP, the cell is producing at rest. There's no demand of energy or something, just to fulfill the normal daily things. And then we asked first, how much ATP is the cell producing? We know from different from years of research and from other groups and from our own data, what is in which range the normal ATP of a cell is. And here you have you see that the normal femtomole per cell is around 1.4 to 1.6 femtomole per cell. That is a normal range you find normally. So now you ask or you look, is the totally ATP less or more? Normally, if it is less, you know that the cell is can't fulfill his normal task, also this normal function. And what most people don't look is what happens if the ATP is too much. I think a lot of people think, wonderful, I can produce a lot of ATP, it's best. No. If it's producing a total of more ATP than normal, you have to ask why. Why is the cell producing more ATP? Sure, first you can be proud of your cells that they can produce more ATP, but again the question, why are they producing more ATP? We did we and others a very simple experiment. If you, from the microbiologists, this is normal test. If you infect your cells with, for example, intracellular bacteria, then the cell must fight this infection. And she's producing a lot of ATP, very, very high ATP. And I think you would not say you are healthy, if you're producing a lot of ATP because you are infected. So first we look at the total ATP. It is in the normal range, too less or too high. So, and the next step is, sure, you know that the ATP can produce in the cell by different pathways. The most important, sure, is the mitochondrial pathway, but you have also the anaerobic glycolysis producing lactate, or there are also other pathways. The cell can produce ATP for emergency reasons, so it can use 2 ADP, and then the cell is generating 1 molecule ATP and 1 AMP. That she can do for a short time, but not for a longer time. So again, we first look at the total ATP, and then we look what is the capacity of the cell to produce the ATP by the mitochondrial pathway, as a mitochondrial ATP capacity. So that should be around always 80% of the total ATP. It must not be 100%. It depends on the cell type. But the cells we are using here, this peripheral blood mononuclear cells, it's about 80%. The next question is the cell able to produce the ATP through the pyruvate pathway, also through the glycolytic pathway? That is the glycolytic ATP capacity. And then is also at last the reserve ATP capacity. So if you, dependence on the results, you can see where's the problem. Is there a general mitochondrial problem, or is there only a problem at the pyruvate pathway? So perhaps you have an insulin resistance, or is there a problem also additionally or at the fatty acids use of for ATP?
Thank you very much. You just mentioned insulin resistance. Dr. Bisogno said last week with evidence that about 95% of the US population appear to be suffering from a degree of insulin resistance. And that's the beginning of glycolysis, isn't it, Professor König, that you need insulin to allow that glucose in?
Yes.
So
yes.
If you have a problem here, letting that glucose in, it says GLUT2 and 4 there. That's actually, the glucose insulin receptor into the cell. Then you'll have a severe problem. So insulin resistance is a big topic in and of itself. The therapists here on the webinar will know that. There are excellent, of course, approaches to reducing insulin resistance, which is often accompanied by metabolic syndrome. This is just one article out of the thousands out there about that. This just gives some nice foods that you can consider introducing, but it's a much bigger topic than that and involves ideally quite a lot of different tests, not just insulin and glucose and HbA1c to see why that might be the case. But your test definitely can give an idea that that might be the problem, can't it?
Yes, and before timely before you can detect insulin resistance by normal test systems.
Of course, yeah. So another reason might be that the cofactors are lacking to get pyruvic acid, which you just talked about, into the mitochondria. And you can see here, this is the pathway, and it's very small on this, so we've increased the size. Magnesium is a vital cofactor, as well as the B vitamins, B1, 2, 3, and 5 particularly, and hidden under this yellow line here, alpha lipoic acid. So those are all vital. And of course, your gut microbes, your microbiome is responsible for synthesizing your B vitamins. So, you know, you may have problems even if you're actually eating foods that contain those if you have gut issues. So that is something you have to think about. But would you back that up as well, Professor König?
Yes.
Doing gut tests, microbiome tests is often very useful when you have a problem here. This is interestingly showing that there is a different pathway for the fatty acids. So we'll talk about that in a moment. The fatty acids may well get into the mitochondria, which is this yellow border here. All of the Krebs cycle and the electron transport chain are inside the mitochondria. And imagine that you have thousands of those in some cells and thousands of these ATP electron transport chains in each mitochondrion. Some people, Professor König, think that there's only one in each mitochondrion because that's what the images show, right?
Yes.
But you've got hundreds and sometimes thousands in each.
Yes. Yes.
So you may find that the fatty acid pathway here is working. We'll see that in a moment, but the carbohydrate one is blocked. And really, each of them should be working well. So natural sources of B vitamins are listed here with all of their references. Just to mention that thiamine, that's B1, has seen a lot of research over the last, I would say, particularly over the last 10 years. A brilliant book has been produced on that called Thiamine Deficiency Disease by Dr. Lonsdale and Dr. Chandler Mars. And they do mention in this article here, for example, that B1 is a critical and rate-limiting cofactor for multiple enzymes involved in this process, including those at the entry points. And that's what we're talking about, isn't it, in the AT profile, is whether you're getting the substrates through the entry points. So I've written here at the bottom, thanks to Elliot Overton. He's a nutritional therapist and a British one, and has covered this in huge detail, talks at the ICANN conferences and so on. So it's really worth looking that up and recognizing perhaps that the benfotiamine, fat-soluble form of thiamine, can often be a lot more useful than just the water-soluble form. And that's something that you in Germany, Professor König, have been using for years. And we barely know here in the UK the form of B1 that is both fat and water soluble, because then it gets across fat membranes and it gets into the brain. So natural sources of magnesium are here. That is vital, even for this glycolytic pathway. And the right balance of electrolytes is important. Interestingly, Professor König, you've talked to me a lot about this. There's a different balance of electrolytes needed in the serum and inside the mitochondria, right?
Yes.
And normally the body can get that balance right, but you have to be giving the sources to the body and then it will choose how much it requires, right?
Yes, yes.
This,
Sorry.
go ahead, sorry.
No, sorry. There's a big difference. You mostly measure it in the serum, but there's a different concentration in the mitochondria. But normally the mitochondria can regulate it, or the cell can regulate it. But sure, you must offer enough of this minerals or electrolytes.
And it's important to remember that what you're measuring in the serum is not what's in the mitochondria, or even often in the cell. Yeah, it varies, doesn't it? But in general, for example, here you can see with copper that while 11 to 30 micromoles are needed in the serum, about 7 times that are often found inside the mitochondria. So that's a huge difference, and you can't measure that. It's impossible. Thyroid hormone is important. Do you want to just say a word about that, Professor König? It is important to do a thyroid panel, isn't it? The link between the thyroid and the mitochondria is huge.
Yes, yes, you see it here on this slide, and you must be sure also that the mitochondrion is producing, but also has a lot of receptors for hormones. So it is essential for the correct function of hormones and production, and therefore especially the thyroid hormones would be worth to measure them.
Yeah.
Include the panel.
And especially the active thyroid hormone T3, which in this country the NHS never does unless you really push them. They'll refuse to do it. So
Yes.
sometimes you need to do this privately to see how your thyroid is really working. So if the mitochondrial ATP on the ATP profile is stronger than the glycolytic ATP, then that's a sign that the fatty acids are getting in, isn't it? Professor König, this is an example of it here, isn't it? ATP capacity for the mitochondria is higher and the glycolytic ATP is lower. I don't know if you want to say anything about this entry portal here.
Yes, therefore we can see whether the mitochondria get the ATP predominantly through fatty acids, or whether there's a problem with the glucose or the glucose pathway. Here, the mitochondrial ATP capacity is better than the glycolytics. Thus, the cell or the mitochondria are preferentially using or can use using fatty acids. Yeah.
Just to remember that there are some mitochondrial fatty acid oxidation disorders that you can be born with. And so it's not just acetyl-CoA that is working here. You know, if this is blocked, then it can be something more serious, can't it? Is this always discovered at birth, Professor König, would you say, in general?
No, I think there are in between there are a lot of genetic diseases which are covered at birth, but I'm at the moment not sure. I think this one is not, it doesn't belong to the tests which are covered.
Yeah, LCHAD, for example, this one here was covered by Dr. Stephanie Seneff at Dr. Klinghart's summit in Brighton that he just held a few weeks ago. Absolutely fascinating. So we do have some information on that if anybody comes across that in their patients. So the mitochondrial health index, this is some markers at rest and a lot are under pressure, aren't they, Professor König? So maybe you could explain which are at rest and which are under pressure. And the fascinating thing is the difference that it shows us in this test here. So this is an 11-page test. So it's not just one page, and gives us a lot of detail. We'll just be showing 2 pages today.
Yes, it's the ATP profile. We have some hints what is whether it's functioning or what is not functioning. But if you have at the ATP profile, everything looks nice, then you can't be sure that everything is also nice at energy demand. Yeah. And if you have an ATP profile which is at rest, not perfect, then you must look where's the big problems, and that you only see with the mitochondrial health index. Yeah, so sure you can start with the ATP profile and look at this one, but the mitochondrial health index gives you much more. And what we are looking, and they are not due to. I was confused because sometimes they are not due to the confidential. Do you understand the chat, Gillian?
No, I'm not looking at the chat. No.
Okay. Okay.
Sorry, I can't see it from here. We will be answering questions at the end, so please put in absolutely anything that occurs to you, and we'll get onto that towards the end.
Okay. So here you see different parts. This is the first page. And I first look at, what I'm first looking is always the potential maximum oxygen consumption rate in picomole oxygen per minute. That is the target, as you see here, it's 22. It's on the bottom of the page. And that is very, very low. So we will look what is able to produce an oxygen consumption rate, or what is in the at rest. At rest is 28 picomole, but on demand it's only 22. So this person has really nearly no energy. Then we look at the cellular energy phenotype at rest and energy demand. And at rest, it should be resting. Sure, it should be used a little bit anaerobic pathway, a little bit mitochondria. But at the on demand, you have also resting. And resting means that the cell can't respond to energy demand. There's nothing going. So normally you should have an energetic or aerobic pathway. That means if you have energy demand, you must take your mitochondria must be activated. And in this case, there happens nothing. And then you can look at different other parameters. You see that the ATP turnover rate is not surprising that the cell is using 100% at rest, and there's nothing anymore for energy demand. And if you look at the coupling efficiency, where we also look, this is the green one, that's okay. So we can be sure that there's no problem with the coupling of electrons. But you have only 22 picomole oxygen consumption rate, so that it means nearly nothing. So in the end, we look, or we concentrate on the non-mitochondrial respiration as a share of total respiration. That's 32%. That is a parameter where we see, or others, in other words, the cell takes oxygen or needs oxygen, and the oxygen goes into the mitochondria, sure. But not only in the mitochondria, there are other cells, the enzymes in the cell, which are using oxygen, and that are oxidases. And what are oxidases doing? They degrade pharmaceuticals. They try to look at heavy metals, or they try to destroy intracellular bacteria. So if the non-mitochondrial respiration is a very high number, we have to think what is the reason. Yeah, Gillian, that was the most important first, and
Okay.
all other parts are some calculations. Yeah.
Yes, I mean, one does need to, Afifa, I've seen your comment now because I did switch to the chat, and you're saying this is such a massive topic. Can we really cover it methodically in the short time? I totally agree with you. It is difficult. That's why we've only chosen 2 tests to focus on today out of the 12 or so tests that we do. There will be much, much more to cover, and we will be trying to sort of synthesize this at the end. So I totally get it that you need a holistic picture. You need to see the patient, right, Professor König? When we talk about these tests with therapists, as we're always prepared to do, we are always happy to do an interpretation of the tests if you do them with us. We always like to ask a bit more about the patient and find out, for example, what could be blocking the mitochondrial respiration, that it's not all going into the mitochondria, because here you can see that only 58% is going in when it could be potentially above 90%. And that's something that this test cannot tell you, whether you have aluminium blocking or Borrelia blocking or Coxsackie. So the next page, page 2, does mention a few sort of top level points that you can go back to your patient with and discuss in greater detail. Would you like to just mention a few of those, Professor König?
Yes, perhaps to extend what you have said, it is normally therapists like to know exactly what to do, which pill should I give, what is the reason. It is really complex, and if we talk with a therapist, we take 1 or 2 hours really to look at all the parameters the patient has, because the one has aluminium, the other has arsen or whatever. So we can't see it from this one. And then we have to discuss with the therapist and look at other parameters also to tell him then what he has to look for. So it's a good direction. Here we look first, is the mitochondrial dysfunction really none, slight, moderate, considerable, or extreme? And in this case, you know it's extreme. 22 picomole oxygen is really nothing. And then we look also in the cellular imbalance. That means does on energy demand, does the cell switch to anaerobic glycolysis, or is still at rest glycolytic pathway, or is it able to, or is the electron transport chain very damaged? And here it is, yeah, it's between considerable and extreme. Yeah, it is in this direction. And then we look at what does it mean, indications of, yeah, sure, increased formation of oxygen radicals in the cell. That is the increased oxygen consumption in the cell outside the mitochondria. In this case, we had no increased formation of oxygen radicals in the mitochondria at the electron transport chain, yeah, because we had no electron, no problem with the electrons. We have a restricted function of the electron transport chain in the mitochondria. Yes, that we have. Function doesn't mean radicals formation. Function is otherwise, if they would function perfectly, we would have a maximum oxygen consumption rate of 100, 170. So there must be a damage. And at this, we didn't measure the number of mitochondria. That is the topic of the next time. So we don't know at the moment whether we have a lot of mitochondria or we have a low amount of mitochondria, but it's indication that we have a limited number of functional intact mitochondria. Yeah, the exact you must order the mitochondrial mass. Yeah, we know from this from the first slide, from the first page, that there is insufficient ATP formation on energy demand. Yes, it's very dramatically. And then we have really to look what are the reasons. Yeah, in our other seminars or Gilian, or if we do it next year, then we have more explanation what could be the reason, yeah, for the problems.
Yeah, we'll just be talking about the micronutrients for now. So this will be a little bit of a sort of rush through many charts,
yeah,
but we can go through this in greater detail in a webinar specifically about the micronutrients another time. So the cell danger response, which is reflected in that non-mitochondrial respiration, is something to keep in mind. In other words, the mitochondria are able to downregulate in a protective mechanism if they see that there's a lot of work that's needed outside them in the cytosol. In other words, cleanup operations. We've mentioned bacteria, viruses, the spike protein, of course, as well, unfortunately, post-COVID, and even remnants of COVID. So I just want to mention that there is an article, I'd actually written that several years ago on the cell danger response. It's available under this PDF link here at the bottom of the page. If you don't want to go and look at the full articles of Dr. Navio's, which are absolutely brilliant, he and his team. And so some of the factors that may be influencing the downregulation of the mitochondria while this cell danger response is continuing, are viral or bacterial infections. We have many webinars on these elsewhere that you can find on our website. Biotoxins or mycotoxins, some parasites. Look at how toxoplasma can tether and disable the mitochondria here. Quite frightening, really. Heavy metals, of course, aluminium, cadmium, mercury, arsenic, as Professor König mentioned just earlier, is particularly pernicious. Pesticides and herbicides, farmers affected particularly by those. Chemical contaminants, household chemicals, EMFs, that's electromagnetic frequency of different kinds, medications. The microbiome may be full of lipopolysaccharides, downregulating the function of your gastrointestinal tract, and that affects everything else in the body too, as you know, and the spike protein from COVID particularly. So this is just a slide showing how dramatic it was when it was first found, how the SARS-CoV-2 virus can downregulate the mitochondria with its spike protein and its viral remnants. It actually blocks apoptosis of those cells that need to be recycled. So that's interesting, and lots of articles about that now. So do consider, the therapists among you, whether that's the first port of call with your patients. You can also measure the antibody, the spike protein in the antibodies of the cell to see whether that's really a big part of the problem. The B vitamins are vital throughout the entire cycle. This is the Krebs cycle here, also called the citric acid cycle. And you can see how many different B vitamins are essential for that. And of course, the microbiome synthesis of those, as mentioned, and the absorption of those, whether you're actually able to absorb them, like B12, through your intrinsic factor. The mitochondria is this yellow line here. Everything that's going on there is inside the mitochondria. Now, the vitamins and cofactors necessary for the electron transport itself are described beautifully in this article by Wesselink, which we've linked to at the bottom of the slide. Do have a look at that. It was also featured highly in the two-day mitochondrial conference just last weekend. I've summarized on the next page the main nutrients that are mentioned with each of the complexes. And you can look at this in your own time if you wish to. Of course, it's very difficult to just give this to a patient and say, well, you'll probably need all of these different nutrients. That's why I know, Professor König, you've spent a lot of time with teams analyzing the different combinations of these nutrients in extracts that can be given to patients, because you can't tell a patient to go out and buy all the foods that contain these and have them when they're chronically ill, right? It's sometimes just a lot easier to take a combined nutrient that contains the cofactors that we're talking about here. So it's like a multivitamin, multimineral for the mitochondria. And maybe another time you can talk about the research you've done into those, because that's what your lab spends a lot of time doing as well, isn't it? Is researching those.
Yes.
Those nutrients. So CoQ10 is critical, and you've all heard of that, but can you tell us a few words about CoQ10, perhaps, Professor König?
Yeah, CoQ10 is essential, no doubt about it. But you also know that CoQ10, also coenzyme Q10, is also synthesized by your body. So you have to ask yourself why to supplement such a lot of high number of coenzyme Q10, or perhaps it's better to see that your body is producing it by himself. But okay, sometimes it's necessary to add it, but it's a single enzyme, and if the other or supplement, or molecule, if the other pathways which are connected with coenzyme Q10 are not functioning, then you can produce sometimes more oxygen radicals. So you are doing not good to add a lot of coenzyme Q10. Yeah, so it's better to add coenzyme Q10 in a combination with other things which regulate again coenzyme Q10, or which takes the oxygen radicals which are generated. Yeah, also it is it's sometimes not good to just take one molecule, yeah, and to forget that this molecule is inserted in other regulations, yeah, or can also be oxidized.
Yes, thank you very much. I do see that sometimes, the sort of 300 milligrams and so on of CoQ10 every day. It's sometimes just too much, isn't it? The body can't process that, and it has to, you know, it creates a backlog.
Yes, you have intermediates. Intermediates of the coenzyme Q10 is oxygen radicals, and if then the next way is not organized, then you have more problems than before.
Right.
Just
so thank you. Yes, absolutely. Food sources of CoQ10. Interesting that that's primarily, you know, meat products, fish, dairy. So if you're a vegetarian or a vegan, it is worth considering how much you're getting and perhaps supplementing. And of course, if you're taking statins. So glutathione, also our key intracellular antioxidant. Professor König, even if the electron transport chain isn't showing reactive oxygen species, you've often told me that this could be the key that's needed in an ailing mitochondrion.
Yes.
For the reactive oxygen species in the cell itself and in the mitochondrion, even if the electron transport chain isn't being affected yet.
Yes, yes. Also glutathione is very, very important.
And so support of glutathione from food sources in this excellent article here, even with the dosage recommended. So that's by Dr. Minich and Ben Brown. Often forgotten is bioavailable copper. We'll probably have a separate webinar about this specifically, because there's so much to say. Just this last ICAN magazine, which is the Integrative Healthcare and Applied Nutrition magazine that's the biggest in the UK, for those who are outside the UK, a very, very influential magazine for both functional doctors and complementary therapists. And the editor has written a special where the cover says copper, the ultimate mitochondrial nutrient, and a very good article. I do have a copy of it if anybody would like that. And a few slides now that I won't go through in detail because we'll have a separate you know, talk on this, I think. But if you'd like to look at those in detail, you'll see how very, very important intracellular bioavailable copper is for the mitochondria. And the iron copper balance is often completely off, and it looks as though the person has anemia when actually it's a different condition. It's inability to ferry the iron around properly because you're lacking copper. Ceruloplasmin is the carrier of bioavailable copper, and it's barely ever included in NHS blood results. And it's every time I measure it, it's low, so often low. Just an example here. This is an article describing how ceruloplasmin is actually an indicator of copper status. And so a few more slides about that we don't have time to go through today. We will another time, but do read them if you can. Retinol, that's the real vitamin A. It's not beta carotene at all. Beta carotene is just a precursor to vitamin A. And that, interestingly, is only available from unfortunately, meat products. It's not, or dairy products. It's not available from plant products at all. So very interesting that you may be lacking in copper without even knowing it if you're not getting retinol, because that's the backbone of the carrier for it. So natural sources of it here mentioned, and references. Would you like to just say a few words about magnesium inside the mitochondria, because that's so important for mitochondrial ATP, isn't it, Professor König?
Yes, but I think this is slide is very, very
it's too complicated, I know.
Yes, it's complicated. but if we just look at the magnesium, and we see that it is regulating a lot. Yeah. So magnesium is just more, just if you have scalps or in your muscles, you need magnesium everywhere in your cell, in your mitochondria. And we in our lab since years, we also know that if you don't adjust the magnesium concentration in your media, then your cells are not functioning correctly. Yeah? So just to summarize, magnesium is important.
Yeah. So the last, that's about one form of fatty acid that's important, DHA. We'll cover that another time as well properly. Do have a look at the work of Professor Crowther from the UK, who's discovered over many, many years how absolutely vital DHA in particular is, and not so much EPA, interestingly, for the mitochondria. So the last test we'll briefly touch on, maybe we need to talk more about that next time as well, is how you can work out what fuels are getting into the mitochondria, the lactate pyruvate index. Professor König, just tell us a little bit about this, please.
Yes, we have again the resting cell and the cell under energy demand. So, and if you have, or if we take peripheral blood mononuclear cells, then we know that they should normally use their mitochondria for energy, for ATP. And you can calculate exactly whether your mitochondria are using predominantly glucose or fatty acids through the mitochondria, or whether they are using glucose through the anaerobic glycolysis. So if you have an index or ratio above 2, as it's our calculation, then we know that the cell is primarily using carbohydrates and preferentially converting them to lactate, so that they're not using the mitochondria. And if you have a ratio between 1.2 and 2, the cell is primarily using carbohydrates and partially converting them to lactate. That means they are converting the pyruvate into the mitochondria. If you have between 1 and 1.2, the cell is also again using carbohydrates, but transporting them eigentlich totally by 100% into the mitochondria. There's not an energy generation by anaerobic glycolysis. So if you go now below 1.0, then you know that your cell is using not only carbohydrates, but also fatty acids and perhaps also a little bit amino acids. And below 0.8, the cell is primarily using fatty acids. And that you calculate for the resting cells and for the activating cells. And in this case, you see that, at the resting cells, you still have, a value of 1.61. That means that your cells are using primarily carbohydrates and partially converting them to lactate. So there's a little bit disproportion, here, and partially converting them to lactate, so around 30%. Then on energy demand, we would expect that then the mitochondria are used because you need more energy. But in this case, it's not true. The cells are primarily using carbohydrates and converting them around 80% of them to lactate. That means there's really a big problem to use mitochondria.
And lactate can be very painful, can't it?
Yes.
Particularly in the muscles. It's what marathon runners feel after their marathon. And of course, in CFS, ME, patients often feel this a lot of the time, and it's a sign of huge post-exertional fatigue, isn't it? Which you can also see reflected in the mitochondrial health index. So just a couple of words about what you can do to assist this, and we'll talk more about it next time. C8 can be particularly helpful if your mitochondria are showing that you're not able to ferry the pyruvic acid into the mitochondria and you're not able to utilize fats, right?
Yes, you can.
C8 is the best ketone, isn't it?
C8 is a good ketone. I prefer sometimes butyrate, yeah.
Beta hydroxybutyrate or butyrate. Yes, there's information about C8 on this page, which you can buy. It's a fraction of coconut oil. So for the therapist amongst you, you may like to. Again, at our conference last weekend, one of the top scientists there said that during fasting, he suggests that his patients take 2 tablespoons of C8, and that almost gives them a feeling of complete fullness and energy. I found that very interesting. But the next page is about the butyrate that you're mentioning.
Yes, and you can test whether you have problems with, for example, with carnitine or with the fatty acids degradation, because then if you degrade fatty acids, then you produce a lot of oxygen radicals, and you need your catalase to destroy all the oxygen radicals, and you need the carnitine as a transporter. And here you can test then whether butyrate is because it goes directly into the mitochondria, whether it can be useful. And here's our normal mitochondrial function test. And in this case, you see that with the butyrate, you get more ATP production. That means the mitochondria is functioning, but you have a problem with your carnitine or with your degradation of fatty acids. Yeah, so you can test whether where might be the problem.
Yes, thank you. Sorry, I clicked onto the next page too fast by mistake, but the problem might be lack of L-carnitine, right?
Might be. Yes.
Acetyl L-carnitine is particularly good. Again, mainly meat and dairy sources of that. So something to consider if you're lacking in energy and you're not taking any of those. Perhaps a supplement would be useful. And the pathways here are explained in great detail. Just a tiny last section about how important it is to protect yourself from EMFs as well, because the mitochondrial energy production is dependent on mitochondrial membrane potential, which is electricity. It's weird, isn't it, to think that it's really the electrons inside the mitochondria that are giving you that energy, and they have to hop along that electron transport chain, that's literally the name, in order to give you that energy. And if they're working well, it's the equivalent of a bolt of lightning. Yeah, that's based on good science. The references are below. But if you've got too many EMFs around you constantly, your body isn't used to that. We didn't develop genetically to be able to tolerate this amount of constant electromagnetic stress. And some people are a lot more sensitive to that than others because of the voltage-gated calcium channels in their cells, which the Wi-Fi actually disrupts. So without going into detail on that, all the different things you need to think about are mentioned both here and particularly in talks that Dr. Klinghardt is giving right now in his updates on Thursdays. So it's worth considering taking part in those too, because there's a lot to say about EMFs. Books coming out actually mentioning that almost primarily as one of the key disruptors of mitochondrial function. So we have lots more on our webpage, aonm.org, and then mitochondria. So please do have a look at the webinars we've had in the past on this. And we'll be talking about the other supplementary biomarkers mid-January in our second part of this. And now I'll have a quick look at the chat, which Brigitte, you may also have already have looked at. I know we don't have long to address the questions today because we don't want to keep you for too long. People have been asking, will the slides be available afterwards? Absolutely, and also the recording. Sometimes they're not made available due to confidentiality. I think, Alessandra, when an article is coming out on the slides, but we don't have slides that contain that kind of information today. what supplements would you recommend the most to help with ME long COVID, lack of energy? Well, Robin, thank you for that question. What we'll do, I think, is make a sort of summary page before we put this out on the web, perhaps, as a chart pack with what we have recommended so far. But as Professor König explained earlier, it does depend a lot on what else is going on in the body. So just this test is often, depending on what parts of it you've done, are not always sufficient to give the full picture. Please could you explain how cellular toxicity, e.g. from heavy metals or mold, impacts the mitochondrial membrane so that fats or substrates can't enter to create ATP? Professor König, could you maybe address that one?
Yes, the mitochondrial membrane, at the moment we don't measure. It is a little bit, we tried it with the chemistry department, because you know, if it depends how the membrane is the composition of the membrane, of the mitochondrial membrane, and then the complexes can correctly build the right complexes to produce ATP. That is not in our mitochondrial health index.
Sorry, it's getting a bit difficult to hear you, Professor König. I don't know why.
Okay.
If you could be, but I think she's asking what the actual pathway is for that. Is that difficult to explain? How for example, aluminium could actually block the mitochondria so that you can't get ATP from it?
Yes, the heavy metals can block at different pathways. But we can't tell it. We can't see at the MHI where it is. Where is the problem? Yeah.
Yeah.
That
Okay.
we don't see.
Okay. Where can we get these tests done? Dawn asks, are they able to be done remotely or clinic only? Dawn, AONM, the Academy of Nutritional Medicine in the UK, is the representative for MMD, Professor König's laboratory. And if you contact AONM, then they will send you a kit. And these tests are best done by a blood draw. So it's just 2 tubes usually, specific tubes for the mitochondria that have a very specific substrate in them. So please don't use other tubes. And then you send them back to Germany. AONM organizes all of that for you, the courier, and makes sure that they get to the lab on time. So they do need to reach the laboratory, ideally within 48 hours. But we have an excellent courier that almost always manages that. It is harder from countries far away like Australia or America or Canada, but from the UK and other countries in Europe, we have no problem at all. And so just contact info@aonm.org and the helpline will give you all the information on that. Perhaps, Claire, you could put those details into the chat. Thank you. What supplements, please, would be best to detox toxins, infections, and precancerous inflammation from the mitochondria? Gosh, that's a difficult one, isn't it? I will be talking on the 22nd of November about KPU, that's cryptopyrroluria, and detoxification needed for that, because it does involve a lot of toxins being excreted from the cells once you replace zinc and sort of get the heme being produced properly again. So Tanya, that could be useful. Otherwise, Professor König, is that something you'd like to address this time, or shall we keep that for the next time?
I think we keep it for the next time. Sure, you can start with alpha-lipoic acid, vitamin C, and with glutathione is always important. But you must know more details of the patient.
And also a binder is important, ideally.
Yeah.
You know, to have something that will actually pull out the toxins and make sure that they are not recirculated via the enterohepatic circulation back up and into the brain, as it were. So that's, you know, either chlorella or zeolite or pectin or whatever. There are lots of ways to do that, but you have to have a really clean binder. Do we have the blood drawn in the UK? Then DHL will take it to Germany. Oh, Alessandra, you were mentioning what to do. Yep, proper history taking and methodical, sensible thinking, and only testing if necessary is hard to teach. These tests are a long way down the road. Thank you, Afifa. I do understand that. It's a rarefied area of physiology. You're right, but it is very, very wonderful that these tests are now available. Since Acumen went down at the beginning of COVID, there has been no tests available. And actually, these are the only tests available in Europe as well. So if one does want to measure the mitochondria, then this is really the place to go. Does the blood draw need centrifuging? No, Lucy, please not, because it needs to be live mitochondria, and centrifuging just takes the serum off, and the red blood cells, it wouldn't be live once it gets to the lab. Anything else to say about that, Professor König? Is there anything that you can do with centrifuged blood?
No, not centrifuge, please not, because we have a separation of cells, a negative separation, and if you centrifuge it, it's not good.
Yeah, yeah.
Because then you have interaction between the cells. Yeah, you have activated cells, and that we don't want.
Yeah, yeah. This is probably the last question that we have time for. Caroline asks, what is the accuracy of these tests? And in relation to the symptoms, could you explain the methodology behind them? Again, big questions. I know you've written over sort of 500 articles and books and brochures and so on, Professor König, together with your teams. But could you just mention anything about the accuracy of the tests?
Yes, it's accuracy. That's classical biochemistry. We did 30 years ago. We had in former times we had the Clark electrode, and we need a lot of cells. In between, we have it semi-automated automatically with the seahorse, and you need only very low amounts of cells. And again, it's classical biochemistry. It's very accurate now, because it's really it's injected automatically. It doesn't depend on your technician. And if you measure it on split samples, we did it. Sure, we validated this. It's the same results. Sure, if you now take your blood and then you do exercises, really exercise marathon running or something else, and then you take again blood, then you have see differences. But normally we really validated this. This is classical. But you can read our papers. It is internationally published. And yes, you can read everything.
Yeah, the seahorse XF, which is used for most of the tests,
Yeah.
though not for the ATP profile. For that, you use other methodologies as well, I know, is used around the world. And we do have a direct link, if you'd like it, Caroline, to over 7,500 peer-reviewed studies that have been done using the seahorse XF. And many, more and more studies are being done with that methodology, and also the way that the mitochondrial health index, for example, is calculated is international. It hasn't just been devised by this lab. I do know, Caroline, absolutely, that Dr. Joe Pizzorno said last week at the EDMI conference that he didn't know of any commercial mitochondrial tests, but he was very interested when I told him about this one. And he asked me to send him details of it. So I will be doing that. And it was just too short notice for him to sort of look it all up and change his presentation and include it. But I was worried by that and did talk to Ben about it as well. But I mean, that's just the way of the world. He didn't know about this particular method and laboratory. So I think that's probably all we have to talk to answer today. But Claire, please do copy over the both chat and the Q&A if you can, because we'll have a chance to answer all these questions either in between now and January or at the next webinar. Thank you so much for being there, everyone, and for bearing with us. It has, it is a little bit after 8 now. I will just mention that on the 22nd of November, we have cryptopyroluria part 2, where I'll be talking in greater detail about the therapies for it, because the first part was just really primarily about the mechanisms. And then in January, probably the middle of January, we'll have part 2 of this webinar. And thank you so much, Professor König, for taking the time to be with us today. Thank you.
Thank you very much.
And do contact aonm.org. Info is your best port of call there. You are very welcome to contact me as well. My details are here. I'm the Director of Research, so I sort of do help to interpret these tests when they come back, but also to choose them if you're unsure what of the tests to choose. The order form is on the website, but we keep the prices absolutely as reasonable as we can as well. So thank you very much, everyone, for being there. And thank you again, Professor König.
Good night.
Good night. Thank you.